Welcome to Introduction to Biophotonics. My name is Marco Molinaro. I'll be your primary instructor along with Professor James Shackelford. We have another student here. Come in. There you are, the last three. Here's your sheets. There's a sign-up sheet going around. This will be, is this, Jim, our 6th or 7th time teaching this course?
Oh, gosh.
The good news for you is that we've made a lot of improvements to the course, and every year it's gotten more interactive. You guys can expect quite a bit of variety in this course. First of all, just a quick show of hands. How many of you are science or engineering related majors? Okay, and how many of you are not? So it looks like, okay, we've got a good mix, a good blend, which is great. We really enjoy that. We love having all those different perspectives. 6 times, okay, this is our 6th time teaching. Really enjoy the different perspectives here. And I'm actually I want to know a little bit more about you guys. So we're going to go through and we're each going to say our name, why we're here, just real brief, and something you did that was interesting over the break. Okay? And I'll start. I'm Marco Molinaro. I'm here because I love teaching you guys, and it's really an enjoyable class. And something fun I did, I got to go cross-country skiing 3 times, which was really quite exciting for me. Normally I only get to go 3 times in a whole season. I got to go 3 times over the break. And my daughter actually came with me and did about 1,000 feet of skiing. And it sounds like, it doesn't sound like all that much. But she's 5 years old, so for me that was a big accomplishment. Last time I had her on skis, she went about 10 feet. So 1,000 feet was a big improvement. If next year we can get her to go a mile or 2, that would be even better, but I'm not holding my breath. But it was fantastic, really wonderful snow. So I really got to enjoy playing in the snow. So
I'm Tim Krabavas. I'm in this class. I really enjoy science, but I'm not a science major, so I thought it was a good chance to take a science class. And over break, I worked a little bit at the Science Museum that I've worked at since I was in 8th grade.
Which one is that?
Chabot Space and Science Center in Oakland.
I know that one well.
Who do you know there?
I knew people
for the old
about
guard.
8 or 9 years ago,
Okay.
But I worked at the Lawrence Hall of Science.
Oh, yeah.
I'm Thomas. I'm an animal bio major, and I took this class because it was a science, but it looked different than your basic chemistry or your basic biology.
You're right, absolutely, very different.
So, and I turned 19 over break.
Oh, congratulations. Happy birthday.
I'm Christina Geddard. I'm a human development major, and I'm taking this class because I liked the presentation with the lights through the finger.
Okay, good.
And something cool I did over break is I actually got to see Northern Lights when we went to Canada, and that was really awesome.
Wow, where were you in Canada?
In Saskatoon, in Saskatchewan.
Pretty far up there.
Yeah.
Wonderful. What colors were they?
They're just white, but they moved, and so.
That sounds really cool.
I'm Lucy. I'm undergraduate in humanities. I'm taking this class honestly because I couldn't get into examine life on the other one. But I'm really interested in Biophotonics, so I hope you don't hold that against me.
I won't, don't worry. No problem.
And something interesting about me is that I saw snow in my neighborhood for the first time. I was in a car driving through the Berkeley Hills, and it was amazing, and I took tons of pictures on my cell phone.
Where was it? Where was it in Berkeley Hills?
Like the Grizzly Peak.
Oh yeah,
road.
Grizzly Peak. I've seen snow there once before, about 8 or 9 years ago. Really cool. Yeah.
I'm Rachel. I'm a mechanical engineering major, and I'm taking a class because I thought that the presentation for it sounded really interesting.
Sounds great. And what did you do? Exciting over the break.
I visited in August.
Okay. So no snow.
I'm Jessie. I'm an NPB major, and I took this class because over picnic day, actually, I saw the presentation, and so I was already really interested in this, and then it came up as an IS thing.
Wow, even before coming here.
Yeah.
Oh, great.
And over break, I'm from the Midwest, so I saw a lot of snow, and I went sledding and skiing and ice skating and on a hike in -4-degree temperature.
So where in the Midwest?
Well, I'm from Illinois, but we have a cabin up in northern Wisconsin, so that's where we were most of the
Oh,
time.
wonderful, northern Wisconsin. Yeah, very cool. Lots of water.
Yeah.
Steve. I'm a civil engineering major. I really like the presentation.
Oh, thank you.
And what I did was I got a
Snowboarding. Great. In the Sierras?
that was good.
Oh, sounds good.
I'm Arjun, and I'm an undeclared life sciences major. I'm in this class because I love biology and I love lasers. So.
Perfect. Perfect.
Over break, I went to Mazatlan with my family in Mexico.
Oh,
And
nice.
the coolest thing was I was swimming and like stingrays were under me for a really long time, and I lived to tell the tale.
Yeah, that's great. Not like, what was it?
Steve Irwin.
Steve Irwin, yeah. Exactly. Great.
My name's Chris. I took this class because it seemed like the most interesting out of the course selections. And I guess the most interesting thing I did over break was play poker on New Year's.
Great. And how'd you do? You didn't bet, right? No betting involved. No. Okay. Good.
I'm Matthew. I'm not really sure which. I'm a civil engineering major. I'm here because of the classes that my schedule seemed like the best one. And my break was on a whole lot interesting. It was just a break, mostly.
My name's Justin. I'm here because light sounds interesting. And I went ice skating over break, which I hadn't done in a very long time.
Where'd you do that?
Some rink near my house.
My name's Nikki. I'm underclass.
What was your name?
Nikki.
Nikki.
I'm underclass in bioscience. I think this class sounds interesting, especially medical applications. And over break, I got to go to the desert outside of Larry Stand Hotel and tour of Joshua Tree.
Cool.
I'm Amanda, and I'm an English major. And I also just kind of got put in the class, but it's all school, so.
Good, good.
And I went to Georgia for the break because I'm from there. But it wasn't that exciting.
Yeah. Well, it was warm, at least a little bit, right? Or no?
It was raining.
Oh, raining.
My name is Katie. I'm an animal science major. I like the presentation for this class, so I thought it was going to take. And I have a lifetime class over the weeks.
A lifeguarding class. Wow. Great.
I'm Caitlin, and I took this class because the presentation was interesting. And over break, I went to the beach.
Which one?
By my house in San Diego.
Oh, okay. Was it warm? Wow, that's great.
I'm Nicole, and I'm undeclared in the ag science major group. And I took this class because I thought playing with lasers was pretty cool. And one thing I did actually over the break, I got a new puppy.
What kind?
Queen Singular.
Wow, very active dogs, right?
Yeah.
I'm Tom. I'm an economics major, and I just kind of got to the class, I guess. And over break, I went to
To Germany. Where were you in Germany?
all over. I have found it all over. So
Oh, that's great.
somewhere else for like every 2 days.
Was it snowy there?
No, it was really, really cold and no snow.
Oh, okay.
Christopher, I'm an international relation major. I'm here because I'm studying exam and life unit.
Okay.
And I want hiking and biking and all sorts of stuff.
Sounds good. How about you guys back there?
Oh, I'm Sean from I'm here because well, I'm getting that in life. But I'm also I'm interested in light in general, so I figured this class on light.
You're going to learn a lot about light. Yes, you will.
And oh yeah, right. I shot a movie over there.
What kind?
Kind of like this strange, convoluted like exercise about acting and very relatively strange things.
Oh, interesting. You may be interested in doing one of our alternative assignments, which involves making a movie for the class. Well, I'll be talking about it a little bit later. Go ahead.
I am Gina. I'm a computer science and double major. And everything was great. I ate cookies.
You ate cookies. Lots of different kinds, or?
I don't know. Interesting to know.
Good, good. Happy to hear it.
My name is Marissa, and I wasn't there for the presentation today, so
So I was just going off by the full paragraph sheet.
Good.
And my roommate said there's a light show.
Okay.
And then,
This is Biophotonics? Is that the class you're supposed to be in? Come on in. Go ahead.
And then so over break, yesterday I made Irish soda bread. I made Irish soda bread in the oven.
Irish what?
Soda bread. Soda bread. And it came out really well.
Excellent, excellent. Happy to hear it. Sounds great. Well, okay, we have one more person. So you're telling us your name, why you're in this class, and what you did over the break.
I'm Sam Glover. I'm in here because this class sounded pretty interesting, and the correct one's being taught.
Excellent. Sounds great. Thank you guys all very much. Before the quarter wears on you, I'm going to take a picture of this group so that I can pass it around next time we meet and get your names written on there so I can have an easier time remembering your names. I'm a very visual person. That's why light and life, all these types of topics work well for me. But I need to get a picture of you guys, so make sure you can see this camera, because I need to see your face in the picture. You can get your name in there. Ready? 1, 2, 3. Okay, I'm going to take one more. Turn off that flash. That's the flash. Okay, can everyone see me? Yeah, I think so. 1, 2, 3. Okay, thank you guys. Okay, so before we get to the fun and exciting things about biophotonics, I just need to let you know that this, my position here in this course is basically funded by the National Science Foundation, which is your tax dollars and my tax dollars going into science, small portion of it getting reserved for education and coming back and funding people like myself in the programs that I carry out and some of the work that James Shackelford also carries out. Because of that, our funder requires us to present them with evaluation data on a regular basis. So I have to give you guys a preset of evaluations. They have nothing at all to do with your grade, okay? Just be aware, really nothing at all to do with your grade. But I need to take a sampling now before you've had any biophotonics, and then a sampling at the end of the course after you've hopefully had quite a bit of biophotonics, okay? So there's two different things I need you guys to fill out. One of them is this biophotonics concept test, okay? There's 14 questions on here. And please do not write on these sheets. There's separate answer sheets that you write on, okay? Don't write on these because there are a lot of color printing and we like to not print these over and over and over again. So you'll be putting the answers on here, and on here you'll have actually it's 13 questions here. Is it 13? I thought it was 14. I thought I was sworn it was 14. It is 14. So you're going to be putting in here question 14. Okay, sorry about that. There's also 3 general questions, and then on the back there's a little question about a graph. Okay, a couple of questions about a graph. So we'll be doing this first. So let me pass these out. Here, if you guys can grab some of these, let me grab half of these, put them in the back. Is that enough for you guys? Here to this course is we're adding an element of data visualization and understanding visualization, which is something that has been missing in the past. And we felt that that was an important element to test out and see if our theories were correct about that. And we'll see after we go through the year, the quarter, and see how that goes. But right now, I'd like to have Professor Shackelford tell you a little bit about the structure of the course.
Okay. You have your handout, which is pretty self-explanatory. And just to give you kind of a broad overview that's not written out here specifically, is that you'll see that actually Marco does most of the lectures in the course. I'll mostly just do a rare lecture. On Thursday, we will include history of science.
Wednesday.
Wednesday. I have to calibrate my brain for 1/4. So on Wednesday, we'll have about half the lecture will be on history of science, and just looking at the two of us, you realize I've seen a lot more history than Marco has, so as a result, I do the history of science part. And for you guys, a few of you were in the Oppenheimer course last fall, which was by and large a history course in many ways. And we'll see some kind of interesting connections to that, in that specifically not Robert Oppenheimer, so much as Ernest Lawrence, whose name we've already seen on some of the slides. We've seen reference to the Lawrence Livermore Laboratory. We have very strong ties to them. Our guest speaker is coming in at 11 o'clock to give you kind of a broad overview of the Center for Biophotonics. Dennis Matthews, the director of the Center for Biophotonics, was a longtime staff member at the Lawrence Livermore Laboratory, and he'll tell us a little bit about how this whole effort in biophotonics here has grown out of that. But in terms of things we'll be doing week after week, there'll be various, it says here, discussions, demonstrations, some hands-on experiments. We'll have a number of guest speakers, and we've really defined this over the years, as Marco said, and I looked up to make sure this is the sixth time we've done the class, and I think we really do have it very nicely polished for you. I think it's a great class, and I think you'll really enjoy it. We have kind of a blend of a lot of activities. We'll have the number of just regular lectures, both by Marco and my little history of science lecture, and then some of the staff in the Center for Biophotonics will talk about specific areas like cancer research and basics of biology. We have an excellent biological scientist who is Marco's associate director of the associate head of the education program. She'll describe fundamentals of biology for those of you who are don't have a biology background, to give you the basic vocabulary and so on. Then, as you see on the back of your handout here, in the description of what's happening in terms of the lab report effort, and it's a little bit different. In the last, I guess from the beginning, we've pretty much focused on term papers, as we do in many of the integrated studies classes. But this time we thought, after getting quite a few term papers, we've archived some of these. We plan to use them on some biophotonics sites in the future, and you'll become familiar with some of those websites yourself along the way. We thought that one thing we haven't done, which is really relevant to the course, and again, many of you are science majors, and those of you who are not science majors, I think it's very good to know how scientific information is communicated. So those of you who are using this as a GE course for science, from a non-science major, you'll have a chance to really kind of get under the hood of how some of these scientific reports are done, and I think you have very good experience. And so we'll have actually two, we have a lot of various hands-on activities, but two kind of semi-formal experiments that will be done. As it says here on the back, one will be on January 12th, coming up very soon. And that'll be one in which you use one of these laser pens here to do a simple diffraction experiment. Diffraction is just sort of the scattering of light, but done in a very special way, where you have a very uniform wavelength in these laser pens. And by having that strike an experimental sample, which would happen to be a strand of hair from your very own head, you'll be able to measure the thickness of that hair. So it's a very simple little geometrical exercise that is actually very high-tech, and it's using a sophisticated light scattering technique that allows you to make a very basic biological measurement of the thickness of your own hair. And then you'll do a scientific write-up on that. We'll show some examples. We have some websites we found which are very compact and very efficient descriptions of how to write a laboratory report. Some of you may have had some basic lab report experience in some of your chemistry or physics classes so far, but this will be a little bit more focused and hopefully a little more thorough experience. Not quite as cookbook as some of those lab write-ups can be in some of your other classes. And in this case, you're going to get a lot of feedback too. One of the things we always try to do in all our integrated studies 4-unit courses is to give you a significant amount of writing experience. And in this case, it's going to be a situation in which you'll also get a lot of feedback on your writing along the way, not just writing one big term paper, getting graded at the end, and sort of seeing if you did well or not so well. You're actually going to get a lot of feedback, as you see from the description here, not only from the instructors, Marco and myself, but also from some peer feedback. All of you are going to make multiple copies of your first report. That'll be read by 3 of your peers, and they'll give you feedback, we'll give you feedback, so you get a lot of input as to how effective the report was. And then you can revise it accordingly, hand in a final copy a few weeks later. And then on February 18th, in the second month of the quarter, we'll do an additional experiment on something called spectrophotometry, obtaining kind of a signature of a characteristic of a particular material you'll be looking at, a particular biological material, and you'll be able to write that up in a report that will be informed by all the experience you've had from the first writing experience. And then you see there's a grand prize of 2 gift certificates to the legendary Woodstock's Pizza downtown, so that you'll be rewarded for, the top reports will be rewarded for their excellent work. And then there's a little list of benchmark timeframes there at the bottom of the second page to indicate when all this stuff is going to be due. And then for the rest of the quarter, you'll have an occasional in-class quiz, so we can kind of give you some feedback on the academics as you go along. And then one of the other big things that we do, we've already talked about the lab reports, but another thing we do that's another characteristic of this particular center, as Marco said, one of the things we have to do because of the substantial funding from the National Science Foundation is do evaluation of the experience that people have from their interaction with the center. You're interacting with the center by taking this particular class. So we would anticipate the exam you just took will be a little bit easier at the end of the quarter than at the beginning when you haven't had a single lecture yet. But aside from that kind of slam dunk improvement in your learning skills as a result of experience in the course, we'll also be doing something the National Science Foundation wants us to do, and that is to do a certain amount of outreach to a broader audience, just beyond the scientists and educators that are involved with the center, but actually to reach out to a broader public. And so we do this in a variety of ways. We have about 4 different audiences, well, 3 very broad audiences that we can interact with. We have historically gone out to a middle school in Woodland, a little town north of Davis. Those of you not from the area may not have been there, but it's sort of a town like Davis without a university campus in it. That's about 10 miles north. And we make a presentation to a couple of science classes out there, usually 7th and 8th graders. And we'll take teams of 4 out. And it's actually a lot of fun. They much prefer hearing from college students about some fundamentals of science than hearing from professors that might go out and talk to them. And so again, we'll divide the group up. We have about 20 students in the class, so there will be several groups of 4, 5 or 6 groups of 3 or 4 as the class divides itself up over the quarter. And usually 2 or 3 of the groups will go out to again, with a middle school. Occasionally we'll go out to a high school class or a few high schools in the region that Marco's been working with. I might want to go out and present to one of those. We've a few times gone over to a community college and done some outreach. We also have usually 1 or 2 groups that will present on campus. I'm sure you've all walked by the quad at various times during the fall quarter and seen there are lots of tables set up around the quad on the north side there next to the MU. At various times, all kinds of clubs and so on, and occasionally kind of an informational booth. And so a couple of our groups have done that. They'll do kind of a Gee Whiz Mr. Science presentation on the quad and have some of the demos that Marco has shown you in the presentation in the fall and show you later this morning about different gizmos and gadgets in the biophotonics field. So you can make those same kind of presentations to fellow students and faculty and staff as they walk by the MU to get lunch. And again, it's quite a bit of fun. And then there are some other options. We very broadly define the general public. One thing that's been done, I think there's a thespian in the class. And we've, last couple of years, we've done, a group of students have done a video. And so, again, we get a chance to present that at the end of the quarter to the whole group. And again, that can be kind of fun. So some people might want to do a little bit of a, I think we have at least 1 cultural studies major and a thespian. So we might have a natural group of, we might have a little production team here forming itself. We'll do that sort of stuff out as the quarter goes on. Then we'll have a final exam, pretty straightforward. We'll show you an example, a final exam from last year. And then the grading is broken down. A certain amount, classroom participation, here a small amount, these regular quizzes that Marco gives out. Your presentations, again, either to middle school, people on the quad, general public, video production, whatever. That's a quarter of the grade. And your lab reports, the writing assignment will be a quarter of the grade, and the final will be another quarter. So it's nicely distributed.
Thank you, Jim. So just to add a little bit about the presentations, what we've had in the past is we've had a puppet show that was videotaped. We've had a dating game that was done about biophotonics. It was hilarious a couple years ago. We've also had a video about biophotonics and people on the street. Somebody just went around Davis and just asked people, what's biophotonics? And we got some hilarious answers. So there's a variety of different ways that you can meet that requirement, and we welcome any kind of creative ideas that you might have. The other thing I wanted to quickly add in terms of schools, we have a whole range of middle and high schools that we can actually go to. We do have quite a few inner city schools in Sacramento that would benefit from having a group of students come in, such as yourselves, excellent role models. And it's quite an exciting opportunity to do that. All the people that have done this have commented that it was a good experience for them. Just want to say something about this classroom participation. 10% might not sound like much, but it actually matters. Okay, it matters quite a bit in the end, whether you're going to get that A minus, B plus, A plus. Okay, that usually makes the difference. What we mean by classroom participation is ask questions. Okay, feel free to ask questions, respond to things, participate. Okay, don't kind of sit there quietly. We actually enjoy having interaction with you guys, and would like to, we want to hear what you guys are thinking about and what's going on with your thoughts about biophotonics. So any questions about this? You can start getting your 10%.
Well, I had a question. I worked at Chabot Space and Science Center, and we had a monthly scientific enrichment. I was wondering if one of the groups could go there.
Yes, no problem.
Yeah.
Going to a science museum would be no problem. Yeah. It would be a great new thing to do. Are they pop quizzes? Yeah, effectively they are. I mean, they're kind of roughly equally spread out. So you can, if you haven't had a quiz in a while, you're likely to be about to get one. They're not very complicated. They're to get you, in a sense, thinking about what you'll need for the final. And what we do is the next session, I go over the results and sort of talk about misconceptions and problems that people are having. So it's a way for me to tell what's going on with the class, what's clear and what's not clear, and also a way for you guys to get some practice for the final.
I just wanted to know what we'd be doing on the field trip to Oak Park.
On the field trip to Oak Park, you're actually going to go to our facility, our Biophotonics facility, and that's where we have, for example, we have the highest resolution microscope, light microscope in the world, commercially available high resolution microscope in the world. We have a variety of different microscopes that trap cells. So you'll get a tour, and you'll also get to image something of your own. So whether it's going to be cheek cells, or you'll take a picture of your hair there, there'll be a variety of different things that you can do. We have some pretty high-end microscopes that you can utilize there and some other equipment, and you'll also get a tour.
Is there a book?
Okay, the book. I've had a variety of emails about the book. The reality is this. There is no introductory book on biophotonics that would work for this class as a whole. Okay? It just doesn't exist. We've been asked to write one, and the reality is it takes a long time to write a book, and we haven't gone there yet. There is a book by Paras Prasad, but it is really a kind of a senior, beginning graduate student level. So if we need to, we borrow some things out of that book. And we can have a couple of copies around available for you to look at. But the reality is you won't really be needing that book. There's things online that are actually more appropriate to the level. Some other elements that we can bring in that would be better. Some other questions back here? So we're saving you some money.
So we need to reference the chapters. Is that in that book?
The chapters that are referenced are in that book. But again, the reality is that book is aimed a bit too high for this course. So if you want additional readings, I'll try to provide some other readings that are more suitable. And actually, we need to write some of those, actually.
Yeah, the bioscience majors in the case, check out that book if they want to, when they've done term papers, a little more quickly in 3D, but generally you don't have to worry about it.
And just I wanted to add something about this peer review cycle. So you will actually write up your lab report, and it'll be about measuring the thickness of your hair using lasers, a variety of colors, and you'll make graphs, you'll talk about your results, you'll talk about errors, all these types of different things. And then you will give copies to your peers, and your peers will evaluate you on some set elements. Okay? So then you'll have a chance to improve your report for a final grade on that report. That'll happen for the first report only. The second report, in which you'll be modeling a finger, okay? You'll actually be modeling a finger with different materials and trying to get it to spectroscopically look like a regular finger. That one you will not have a peer review cycle. Okay? You'll just do it and we'll grade it. And that'll be the one that we use for the Woodstock Pizza certificate. Okay? Any other questions about the structure of the course? Yes?
This report is worth 12.5%.
You know, I think it will be. I mean, in case I'm not going to guarantee that, but it'll be roughly that. I mean, it may end up 10, 15. That's the thing I don't know yet. It depends on how you guys end up doing on how things are looking on the first one. Since it's the first time we do the lab reports, I kind of want to reserve that option to change that. Okay, so I'm going to give you a quick 5 to 10 minute intro on biophotonics, and then you're going to hear from the director of the Center for Biophotonics, Professor Dennis Matthews, who's in the back right there. And he'll give you kind of the overview of the whole center. But first I'm going to give you some real basics. Okay? So, practical biophotonics. Pretty much everything in biophotonics has a light source, a subject, and in our case it's a living subject, a sensor that sensed how the light interacted with that living subject, and some way to interpret that interaction, usually through a computer of some kind. Okay? Now, those of you who saw this, I'm going to turn down these lights. You guys see the two lasers? Which one looks brighter to you? Okay? Now, if I put this green laser on her finger and touch it like this and turn it on, what will we see? If I turn it on, what are we going to see? What color? How much? It's the green one.
Right. You're going to see nothing. No transmitted light.
There we go. What do we see?
Reflected light, not transmitted light.
You see anything?
A little transmitted light.
A little bit coming in, a little bit on the sides. You guys see it? Just a little bit. And here I'll walk around with my pinky, the thinnest finger. You can see a little bit coming through. Just a little bit. A little bit. See it? Okay. Now what's going to happen with the red one? Remember, this is the red one. What's the red one going to do? What? It's going to go through? A little lot? Hello red. Here we go. Right there. Okay? Now why might that be, guys? Why might that be happening? Why is the red going to go through? First of all, what's in my finger? Bones, blood.
Lots of water.
Lots of water. What else? Skin. What else? Carbohydrates. Okay, come on. Proteins. What else? Fingernail. What? Chlorophyll. I doubt it, unless I've been scratching plants. Probably not. No chlorophyll. But I know if it was a plant, there'd be chlorophyll. What else? What? Fats? Yeah. Unfortunately, yeah. No, actually, fortunately, we need the fats. Sorry? Melanin? Sure. What else? What makes you who you are? DNA and its counterpart? RNA, okay. So all those things absorb some light, reflect some light, some of the structure scatters different colors of light. All of them vary. Every single one of them varies, okay? So it happens to be that red and near infrared, like your TV remote control, can go through the most. The other colors don't go through a lot, but they go through a little bit, because you saw a little bit of the green coming through. If I take a white light and stick it on my finger, what are we going to see? We kind of see a reddish orange, okay? But this is, in a sense, proof. And by the way, when you move it down, you can sometimes see the some of the vein structures and things like that. It's kind of interesting. It's almost like a very simple X-ray. What happened to the other colors? They got absorbed. Okay? So a lot of what we do in biophotonics is we take advantage of what happens to light of different colors that go through tissues. And how do we take advantage of that? Well, we do things like this. Here, go ahead, stick your finger in there. You guys can pass this around and try it. What this little device does, you gotta wait for it to blink green, and then you gotta read the numbers. What numbers are you guys getting?
99 over 74, 98 over 76.
98, okay, 99. So 99% of your blood is oxygenated. That's good. And your heart rate is 84, 85, so you're getting more excited because you get a nice result. Okay, how about you?
179.
179, there you go. So your blood is fully oxygenated. All your hemoglobin has oxygen. Very happy hemoglobin. Go ahead, pass it along to these guys. Okay, now how does that thing figure that out? It's figuring out my heart rate, and it's figuring out how much oxygen is in my blood. How can it do that? What is it doing? What is it doing? What do you guys see there? Thank goodness this is not a laser. This is actually an LED. Otherwise, this would be illegal, what I'm doing right now. I can't point a laser at you, okay? It's actually a misdemeanor in California, believe it or not, because of all those high school kids that were shining lasers at each other. You guys probably, a couple years ago. Okay, do you see the red LED in there? Okay, see? It goes through my finger, and on the other side, do you guys see what's on the other side here? What's on the other side? A little sensor, okay? A little sensor. So I have a light source. What's my finger? The subject. I have a sensor, that little box on the other side. It senses how many photons, how much of the light goes through my finger. And then what's the interpreter? Little electronics with a little readout right over here, okay? Okay? That's the basics of pretty much all biophotonics instruments. Now, this is actually two different colors. There's red that you can see, and there's a near infrared, just like the TV remote control. Later on in this quarter, you'll get to actually see that because we'll have instruments that can detect that. And we'll see things that your eyes can't see. And we'll see that there's actually two wavelengths here. And we'll actually see that the way this thing works, it turns on the red, turns it off, turns on the near infrared, turns it off, has nothing, and then starts the cycle again. And it does that cycle about 120 times a second. So it does it very quickly, okay? Very, very quickly. And because of variations in your blood oxygen, in your breaths, between your heartbeats, it can figure out your heart rate too, okay? That's how this thing works. Now we have the famous brain size measurer. Here we go. We swipe it across your head, and we can tell how smart you are. No, we can tell that you are getting close to having a fever. 99.3. You're not quite there. You're still okay. Okay? Let's see who the hottest person in this room is. 98.8. Okay? If you guys want to try this, you got to put it on in the middle of your forehead, press the button, hold it pressed, swipe it across, and then let go. And then you can take a reading. Okay? Now, how does that thing work? What's the light source? Any other ideas? Fluorescent lights is one idea. What's the light source?
Your body.
Your body, how?
The infrared heat that your body gives off.
We are all light bulbs in here. Okay? Every single one of you is giving off electromagnetic radiation. You're all glowing. Okay? And today's not a day when you're going to be sweating. So you're all glowing in here. And the thing is, you're not glowing in a wavelength that if we turned off all the lights and if this room was perfectly black, I couldn't see you. Because my eyes can't detect the far infrared that you're emitting. But that little device can. And it matches it to something called a black body radiation curve. And that curve is very, very, very sensitive to temperature variations. It's temperature to the fourth. Okay, so a small change in temperature makes a big change in what this senses. That takes 3,000 readings in the time you swipe it across your forehead and figures out the highest temperature it measures. Now, it's not measuring the temperature of the surface of your skin, what is it measuring?
The intensity of the infrared light that your forehead gives off.
Not your forehead. What's body temperature?
Basically temperature for blood, right?
Well, is it the temperature of my blood in my pinky, in my little toe, in my heart, in my stomach, under my armpit, in my ear? What is it? What's body temperature? What? That's another place we measure it. But what is the definition of body temperature?
Average.
Is it the average across my whole body?
Or it's core temperature.
Core temperature. What's core temperature? And here, pretty much my heart. But it would be really uncomfortable to have a big thermometer stuck in here touching my heart, right? Really uncomfortable. So we want to measure the temperature of the warmest blood we can find. Okay? Here it's kind of buried in. It's kind of hard to get to. Okay? Things get to the surface, but still it's lost a lot of the temperature. Okay? It's not as warm. So what is the one organ in your body that's incredibly hungry for fresh blood? Your brain. Okay? So your heart beats, blood gets pushed this way, the temporal femoral artery gets shunted up into your brain to feed your brain, and it goes right behind the ear and up here and feeds it. You guys seen those people that get angry and they've got we call it the bulging vein? It's actually a bulging artery. Okay, it's not a bulging vein, and it's the temporal femoral artery, and that's what that thing's measuring. So why do we take it from here all the way back to here? Because every single one of us is different. Some of you have it here, some of you have it here, some of you have it here, some of you have it here. I don't know where you have it. Okay? And that variability is actually why those don't get used at the doctor's office very much. Okay? They're as accurate as an anal thermometer, much easier to use. I wouldn't have gotten any volunteers here to do that.
Why wouldn't you do it on your neck?
Why wouldn't you do it on your neck? You can do it right here too. This is a good spot to do it, actually. It's quite good. If you're sweaty, this is a good spot to do it because this doesn't tend to be as sweaty as here. Yes?
Is that what the ear thermometer is doing as well?
The ear thermometer is basically doing the same thing, but the thing about the ear thermometer is that everyone can stick something in your ear. This one requires you to actually have some skill in utilizing it. That's why this isn't used in most doctors' offices, because they're afraid that a person will take the measurement wrong. In the ear one, they just go, they stick it in there. Now, the problem with the ear one is it doesn't necessarily, it isn't quite as accurate. It's close. But again, there's variation, right, with where that temporal femoral artery is. In the ear, you're kind of bound by this little area, whereas here you really get to sample the whole spot, and actually it's a little bit more to the surface.
What was the button you?
The button?
Yeah, like when you stop pushing it, then does that take the reading right back place?
No. What it's doing is when you first initiate the button and you swipe it across and then you let go, it's taking readings during that whole time the button was pressed and finding the highest one. It's measuring the highest temperature. That's why we take it from here all the way across. Okay? So those devices are like $30 at Costco and are a boon to parents of young children like myself. I have a 5-year-old and a 10-month-old at home, and 10-month-old, this you just go like that. It's very fast. Anything else just will not work all that well, especially with that level of accuracy. So that's another device. And here's the last device we're going to talk about today, though you're going to be seeing many more devices throughout the course, throughout the quarter. This device right here shines 2 different colors of near-infrared. So again, near-infrared, it's like your TV remote controls. So it's like taking 2 different remote controls that have slightly different frequencies, slightly different wavelengths of near-infrared light. We shine it on your dominant writing arm, and we can figure out your percentage body fat. Okay? This one, if you want, it takes a little bit of time to set it up. If you want at the end of class, I'll be happy to show you how this one works exactly. But very simple, not quite as accurate as dunking you in a big tank of water, but again, much easier to do. And one of the big things to think about with biophotonics is that they tend to be tools that are non-invasive, meaning I didn't have to poke you, prod you, cut you open. And they tend to be quick and simple, and they use light. Okay? Now the big thing for you guys to start thinking about when you hear the word light is that light is not limited to the colors of the rainbow. Okay? We're going to be talking a lot about ultraviolet light, which you've probably heard with in association with getting sunburned, but also many other ways in which ultraviolet light is used. We're going to talk about visible light, and we're also going to talk about near infrared, like your TV remote control, all the way out to far infrared, which is what you give off your body heat. Pretty much everything that consists of or is made up of vibrating molecules. Can anyone tell me something that might not be made up of vibrating molecules?
Something at absolute zero?
Something at absolute zero, the molecules would be stopped, yes. But pretty much everything that we interact with all the time, okay, every day, the floor, our bodies, the wall, everything around us, okay, is made up of vibrating molecules. When you're made up of vibrating molecules, you give off electromagnetic energy of some kind, somewhere in the electromagnetic spectrum. And there have been devices created to measure that electromagnetic radiation. Okay? We're going to be mostly interested in ultraviolet, visible, infrared, but we'll have a few forays outside of that range. Okay? So now I'm going to turn it over to Professor Matthews. So I'd like to introduce you to the director of the Center for Biophotonics. This is Professor Matthews, and he has made the center possible. He was one of the lead people in writing the original grant that made the center possible, and we've been around for, now we're in our 7th year, and he'll be telling you some basics about biophotonics and the center in general.
Okay.
And I think also science in general.
Yeah, I'll try to talk a little bit about anything that comes to mind. Welcome to the class, and thank you, Dr. Molinaro and Dr. Shackelford, for allowing me to lecture to you. I've just spent about 2 weeks in Tahoe, watching it snow every day and ski a lot, so I think probably like the rest of you, it's kind of hard to actually have to come back and work today. It doesn't show it too well here, but I have a little astrocyte here that's labeled. Somebody mentioned fluorescence earlier, so Dr. Molinaro has already probably acquainted you with some of that, but I'll tell you a little bit more about it as we go through. But that's one of the powers of what we do here, and we can look at it from different angles. I apologize, our projector doesn't have enough dynamic range, but you can see perhaps there the little rotating astrocyte that we've labeled in different places with different colors. So if we want to know something specific about inside of a cell, a living cell, a neuron, or whatever, we can label it with different colors at different locations or at different points of interest, different organisms or suborganisms, if you like, within a cell. And that's one of the powers of biophotonics. Our center also specializes in education, and you're part of those education programs, and Marco is responsible for all of them. And most of all, we like to build a lot of new technology, and when you go, I'll show you some today, and when you go on a field trip out to our main headquarters in Oak Park in Sacramento, you'll see a lot more. So what is biophotonics? Marco again has already given you a pretty good introduction to this. I'll just mention a few more things. Really, it's a combination of the science that investigates the fundamental interaction of light with matter, you know, lasers interacting with matter, looking at light that fluoresces in different areas of a cell, for example. And it's the technology that utilizes that light to observe and or manipulate biological systems. And we've coined this phrase over time called shedding light on life. It's kind of a play on words. You'll hear about our field in different ways. Sometimes it's called biophotonics. That's the phrase that we've coined. We like that one a lot. Other people talk about bio-optics, again using the photon technology through optics, and or biomedical optics. But as far as I'm concerned, these are all the same. So Marco also mentioned what part of the electromagnetic spectrum we use. This is the entire spectrum, everything from radio waves all the way out to gamma rays. We tend to work inside this red box here, which is sort of the far infrared, all the way out into the hard X-ray. And in principle, and mostly we work around the infrared and ultraviolet region, but we're very greedy about using photons. We'll use them anywhere we can and at any photon energy that we can, mainly because it helps us see stuff, or helps us manipulate biological matter, as I talked about on the previous slide. So some examples of biophotonics, and we tend to break them up into imaging and sensors and medical devices. So imaging devices here, we've labeled, this is a living animal, it's a rat, and we've labeled, if you like, a tumor in this particular rat inside of its brain. We can see into that region using something called bioluminescence, the same thing that makes fireflies light up. I know that you poor, students who live in California don't really know what a firefly is probably, but other parts of the country you can see these animals or these little insects that fly around and they light up, and it's a chemoluminescent or bioluminescent reaction that makes that take place. And we take advantage of that too. We can label, if you like, specific sites inside of a living animal and look in the light that comes out of them. If the animal is small enough and got clear enough skin, clear enough meaning that the light can get through it, that we can actually observe where that tumor, in this case, is located. We also work with nanomaterials. These are basically vials filled up with quantum dots. And these dots luminesce. They give off light in proportion to their size. Actually, it's inversely proportional to their size, but the wavelength of the light. Anyway, we can use these to, again, label, if you like, living animals. We can use these at any wavelength we want because we have control of their size. They're controversial, however. Most of them are poisonous. So we've been trying to figure out for some time now, scientists and among our own and other places have been trying to figure out how to make them not toxic. And we coat them with some materials to make them obviously safer to use and so forth. But they are gaining acceptance, but slowly, as far as using them on animals as sophisticated as ourselves, we still use them on animals that we can sacrifice in the laboratory. This is another example of an imaging device, and this is a microscope image showing one cell. In this case, it's a T cell, human immune cell, that has been infected with HIV. And this cell is transferring the HIV virus from itself to a cell it has come into contact with. This is a very important observation because I'll show you a movie of this a little bit later on, but we're actually learning how cells infect other cells. And the power of biophotonics, and if you don't get anything else out of this class, I want you to get this one thing. We study living materials. We don't want to look at stuff that we have killed and sliced and put under electron microscopes and so forth, because life is very dynamic. It's always changing. So we want to understand how this process takes place, because if we really understand this, we can figure out how to block it. We can figure out how to make one cell not infect another cell. That means you can cure infections, and most notably, could we cure the transfer of HIV? Sensors and assays. Marco passed this around. You've already been working with the pulse oximeter. Again, he's going to probably, I won't spoil his class on telling you how that works, but anyway, that is clearly a very important sensor. If you've ever been into an emergency room, practically the first thing they stick on your finger when you show up there is this device. It's so incredibly powerful and ubiquitous. This is what's called a gene array, sometimes called a protein array and so forth. Again, it's taking advantage of fluorescence labeling of genes or proteins. This is how drug discovery takes place. It's the very heart of drug discovery. They have 96 well, 148 well, all kinds of well, little cuvettes, micro cuvettes. They put drugs in them. They have living cells in there. They see how they interact with those drugs. And the color that lights up tells them what kind of reaction is taking place. Is the drug harming the cell? Is the drug not doing anything to the cell? Is the drug nurturing the cell? And so forth. So this is really, really a powerful device for creating the pharmaceuticals that we have today. This is another example of a nano sensor. This is like the nanoparticles, only in this case we've got a little bead, very, very tiny. It's only about 400 nanometers. It's inside of a Chinese hamster ovary cell, and we can use something called optical tweezers, which I hope you'll get to learn about. But it's the ability to take light, laser light, shine it on a very small particle, and the laser light has enough momentum, it can actually move that particle around. And that's why we call it tweezers. We can grab a particle and move it wherever we want. And so in this case, we can move it inside of a living cell. But that's not all. We've made this particle, we've functionalized it, we call it. We can look at a spectrum of light that's emitted by that particle, and we can tell what, in this case, is the pH, or the local acidity, or base level of, if you like, that part of a cell. So I can tell if the cell is too acidic here, too acidic there, wherever I want to. And I can measure a lot of other analytes in the cell. What's the local glucose level, for example, inside of a tiny cell? And that's important for right here in drug discovery, because I need to find out how is that cell reacting on the inside while it's alive to some sort of an insult that I give it, either by feeding it the wrong food or by giving it a drug. And the last category, of course, and the one that we try to use the most from our fundamental knowledge going to the practical end of things, is clinical diagnostics and therapy. This is a picture of my co-director of the center. You can't see him here. Hopefully you'll get to meet him later in the course. But this is Jim Bogan. He's a neurosurgeon. And so Jim is using a laser, in this particular case, to perform neurosurgery. He's actually excising or cutting tissue out of someone's brain. And that's a very, very important application, obviously, because when it comes to your brain, those cells are very valuable. And you don't want to cut out the wrong ones, number one. And number two, you want to cut them as precisely as possible, because, again, you don't want to lose vital material inside of your brain that either leads to memory loss or inability to move your arms or blink your eyes or whatever. This is another example of using a laser, only in this case we were cutting tissue. In this case, we're allowing light to initiate a chemical reaction. And this is called photodynamic therapy. In this case, a colleague of mine is shining some red laser light on a person who has cancer. And that person has been injected with a drug that is photosensitive. And when it interacts with that drug inside your tissue, interacts with light of the proper wavelength, it becomes very, very reactive. And it kills cells where it has actually been assimilated. So the drug assimilates primarily in high metabolic activity tissue, which is what a cancerous tumor is like. Shine this light on it, it then kills that tumor. It's a very interesting phenomenon. It's FDA approved, and they use it now. It's very important to treat areas like your esophagus, and your stomach, your colon. Many, many areas like that have been treated with this type of device. This is an example of where we're going. Our ability to not only image bad tissue, cancerous tissue inside of normal tissue. Here's normal, here's diseased tissue shown in this region. But then through the power of the laser, and we're working on lasers that have extremely precise cutting capability, they can actually go in here and poke a hole in a single cell. And they certainly can saw, if you like to use that term, they can blast out a few cells with inside of normal tissue. And that gives you the ability not only to do extremely precise surgery we call it on the precision level of a single cell, but I hope someday it enables us to get away from having to use chemotherapeutic agents. These Byzantine pharmaceuticals, which seem to work in some cases, but they basically poison the entire body, and hope because cancer cells are extremely hungry that they'll eat the most of the poison and that the rest of you doesn't die in the process. I think we need to be a little more precise than that. So that's why we want to go in and actually saw out, if you like, cut out with light a single cell at a time, a single diseased cell at a time. So this is kind of a grand overview of biophotonics. There's many, many different facets I haven't shown you. More examples are shown here. Again, the beauty of being able to label different elements inside of cells with different colors. These are all different kinds of cell types. The ability, in this case, we're looking at fluorescence light. We shine in light of one color and look at light that comes out in another color. It's sort of continuous in time. In this case, some work a colleague of ours called Paul French is we send in light, this very short pulse, and we look at the lifetimes of the light that comes back out. And the difference here is that we have to label most of the time these elements or these organisms with an external agent in order to see the fluorescence. In this case, we just depend upon the fact that different tissue types emit light that, if we fluoresce it, that lasts different periods of time. So it gives us a very powerful way of labeling things. This is another example of something where you use, it's like the pulse oximeter. In this case, they're shining light through your skull. It's actually able to go through your skull and back out. Not very many photons make it, but just enough. And the long wavelength ones, as Marco showed you earlier, the ones that tend to dominate in going through the skull. And what they're doing, just like with your finger, they're measuring the hemoglobin content in the upper levels or the outer regions of your brain. What is the oxygenization level there? And so this is, for example, used with babies when they're being born, intrapartum. Are they in fact suffering some kind of strangulation that cuts down on the oxygenization level in their blood, which of course leads to some long-term birth defect? Or in fact, when people are having heart transplant or open heart surgery, where they stop the heart for a while, they put them on a heart-lung machine. Previously, they've had a lot of trouble with this because people wake up and they don't remember their relatives, they don't remember what they do for a job and so forth. It's because they've had some neural damage. Tends to go away in time, but not always. In this case, they can monitor it much more precisely, looking at blood oxygenization level and using biophotonics devices to do that. A lot of other things, these are lab animals again, that we can measure different sizes of tumors in over a period of time. And this is great if you're a lab animal, because it used to be I would grow a rat for 12 days or a mouse for 12 days and then kill it, and I'd cut it up, see how big the tumor is. I'd grow another rat for 14 days, and I'd kill it, cut it up, and measure the size of the tumor. There's problems with that. One, eats up a lot of rats. Maybe that's not something that bothers you. But number two, it presumed that tumors that have been started at the same time in a cohort or a group of rats will grow at the same rate. I would rather observe this in a living animal, and that's what this technology enables us to do. This is bioluminescence, showing a tumor growing, growing, growing. It's the dark or the bright green region that's growing. And now I can give that animal a drug and see if that tumor shrinks, continues to grow, or whatever. And I've done it on a single animal and watched it occur over time. So metabolic activity is preserved. And these are my all-time favorites. Down here in the corner, because it's going to pull your eye to that, is the idea of looking at living animals. Now this is a zebrafish, and this is using something called optical coherence tomography. It literally allows you to do histopathology, which is what people do today, pathologists do today. They cut a piece of tissue, they call it a biopsy, they stain it, they smash it out and so forth, and they look at it under a microscope, and they can tell a lot about tissue morphology. Is it normal tissue? Is it abnormal tissue? Optical coherence tomography shoots light inside the tissue, and then it reflects on different surfaces and comes back out, and it's able to image that. And that's what you're seeing here. You're imaging into this living fish, and you can see the surface, you can see inside of it, and you can see its heart. And its heart is actually beating. So you're getting an image. It's like an X-ray image, only it's done with optical light on a living animal. There's another favorite of mine, by the way, I want to call your attention to. I don't know how many of you are like me, but whenever they start trying to pull blood out of me, it's not much fun. And they get 3 tries, usually 2. On the 3rd try, I pass out because they haven't found the vein yet. So there was a company that came to see us. At the time, their name was called Inframed. And they said, look, we want to image veins inside of your arm. Now, I think Marco will probably do some work with you later on this. But it turns out that your veins, because they have a different scattering power for light of a certain wavelength, infrared light in this case, will provide contrast compared to the surrounding connective tissue. So we're using this, of course, to develop the capability for, this company is now starting to sell a product, helping a nurse, in this case called a phlebotomist or affectionately a vampire, who can go in and actually find your veins, pull out the blood they need for whatever samples they're doing and so forth. This is a real problem, especially for pediatric patients, small patients, young patients, and also for people who are obese. So it really speeds up the time it takes, or shortens the time it takes to draw someone's blood. And then my all-time favorite here, and this is from our own work here, some graduate students and professors here in our Center for Biophotonics. Again, as I told you, the message is we want to be able to see living things. And this is a whole array of cells that are in contact with one another, and they're T cells, and some of them are infecting and some of them are not. So we can actually study this all in fluorescence and dynamically on living cells. Now we sped this up, to be honest with you. This takes hours for some of this to take place, but nevertheless we're able to see what goes on when one cell spreads infection to another, and we can do that over a cohort of many, many cells. Biophotonics goes on all around the world. Our center, shown here in the Center for Biophotonics, is by no means the only one. There's great other institutions in California. UC Irvine has one, the Beckman Laser Medical Institute. If you read Russian, there's some great institutions over there. I've spent a lot of time in China. Asia has a great deal of them too. So it's a great field to get into. If you want, you can see the world, a lot of great places to work. Marco and his colleagues actually put together a whole website on it called Biophotonics World, where we try to put together who's doing what, where in our field of biophotonics. And if you have a chance, I urge you to have a look at that. You'll see many activities going on around the world. It's just www.biophotonicsworld, all one word, .org. So biophotonics market. I show you this because it's a big market. You probably could guess that already from some of the things I've been talking to you about. But if you look at different sectors, non-medical applications like biometric devices, which is where, for example, they can scan your finger, determine if you are who you say you are, scan your retina, determine if you who you say you are, and so forth. Testing components, a lot of probes, molecular biology agents, and so forth, fiber optic sensors, medical therapeutics, and medical diagnostics. And some of our graduate students put this together a while back in School of Management and Business here at UC Davis. But 53 billion and growing, actually. Some estimates are as much as 30%. So this area got gold stars on it. It's what I call theranostics. Diagnostics and therapeutics, I sort of conjoined those words to come into theranostics. And that's the area of largest growth and I think has the largest impact on society. And we'll talk a little bit more about that in the remainder of my lecture. A lot of companies here. I only show you a portion of them that we've surveyed, about 250 of them. We have a database of all these companies. So if you're interested in finding jobs when you get further along in your career, we'll come see us and talk to us about this. Again, breaking things down in imaging and microscopy, sensors, assays, and probes, and medical diagnostics and therapies. You can see we've gone from sort of the fundamental science part with biophotonics. The field goes all the way up to the medical diagnostics and therapies, and many, many different things, some of which are highly controversial. So there's something called hair max, which Marco and I'd like to use. Maybe Marco still has one of these, I don't know. We've been threatening to buy one, but that's this idea. It's actually FDA approved that you can use low-level laser therapy, which in this case is infrared light, I believe, and you shine it on your scalp. It's in the form of a hair. This might be in that heat, local heat, which could be generated by the infrared light, or even local nerve stimulation, could in fact stimulate again in increased perfusion of the scalp. And if you can increase the perfusion, that's going to, of course, lead to enhanced hair growth. Personally, I have some skepticism about this, but nevertheless, they have gone through some clinical trials. We are working with some other people, by the way, I think more seriously, looking at laser light as an agent for therapy of pain. And again, it's the same concept. When you take aspirin, all you're doing is systemically increasing your perfusion. And that, of course, can reduce inflammation, reduce pain. If I could do that using a local, in this case, stimulant, using laser light, it might be very, very effective, especially if I could use it not only increase perfusion, but actually block some nerve impulses that are indicative of pain. So we're working with some people right now to do that, but stand by on how that works out. So current biophotonics work at our center, finally getting to that point. This is our headquarters building in Sacramento, Oak Park. It's right across the street from the medical center. It's a nice big building. You'll get to see it as part of your field trip. We are inherently multidisciplinary and multi-institutional because we figure that this field, taking basic science, fundamental interaction of light with matter and going all the way to medicine, is not something one individual or even one discipline is going to be able to do. So we tend to work with people who have different skills than ourselves. And also we like to work on teams because we've figured out for some time now that the big problems in science now require, most of the time, more than one single genius. We'd like to have a whole team full of them. Our center has about 120 PhD and MD researchers in it. We've educated about 250 students to date, undergrad and grad. At any one time, we have about 30 projects going on. Our partner institutions are shown here. UC Davis, I don't know why I can only find the water tower for its scenic landmark, but anyway, UC Davis, UC San Francisco is the lead institution, UC San Francisco works with us, UT San Antonio and so forth, even Lawrence Livermore National Lab, Stanford, Berkeley, and many other institutions actually that aren't even shown here. We work on a variety of different projects, again broken down into this advanced imaging and spectroscopy, molecular and cellular biophotonics, which we tend to do basic research with and also sensor development with, and in the medical biophotonics. This is a lot for you to digest. I just want you to know that we're working on very high resolution microscopes that go well beyond what you could see or buy today, well beyond the dimensions you could see with what you could buy today, even down to imaging single molecules and a lot of label-free techniques. We engineer probes, labels that we can use. We study membrane proteins. We understand viral infection. I already talked to you about that. And we build a lot of different medical devices and even new kinds of assays that are based on optical techniques. I'll show you just one or two of our most famous technologies we've been developing. Then I want to end up with little projections about where I think we can go with biophotonics, and I'm hoping some of you will join in on helping us get there because it's not going to happen in my working lifetime anyway. So this is my favorite cartoon of trying to understand cellular behavior. I don't even remember who I swiped it from, but there's his name, so I gave him credit. Kind of even looks like Obama, doesn't it? I don't know. Copyright BMA or something. Anyway, so the goal here is to push the imaging capability of optical light down into the regime of X-rays. All this region down here in the angstrom or 0.1 nanometer dimension size, the actual size of atoms and molecules and so forth, has heretofore been dominated by using electron microscopy. And remember I told you earlier, electron microscopes, like before you can use them, you got to kill stuff, freeze it, slice it, stain it, do all kinds of things in order to get the contrast going. We want to take what you can do with optical microscopy, which very much studies living things, bacterium and so forth, and push it down into this regime. And obviously we have some ideas about how to get there: structured saturated illumination microscopy and X-ray, FEL, diffraction microscopy. So the question is, can we equal the resolution of the microscope? And we definitely think we can. And why is shown here. This is and the National Science Foundation held a contest earlier it was last year, coming up with the best scientific images to put on various places. And this was the first prize winner, Science and Engineering Visualization Challenge, NSF and the Science Magazine. Graham Johnson put this together. But the reason I bring this up is we tend to take our technology and go find out who needs it. We call ourselves end-user driven science. So one of our good colleagues from University of Laval in Quebec City in Canada, Yves de Konik, he's a neurobiologist. And every time we think we know how to do stuff with our fancy new imaging stuff, imaging equipment and so forth, he slaps us back down because he reminds us of how complicated the problem really is. So this is a picture of a neuron. And I don't know how much intricate detail you can see here, but the point is to image a living neuron in your brain or in some other part of your body where nerves are critical. You need nanometer resolution, the ability to see things over a nanometer scale size, over millimeter dimensions, because these neurons can be a millimeter or more long. So think about that. Think about it just from the image acquisition point of view. Think about it just from how many pixels that's going to be, or how many megabytes that's going to be, in this case, petabytes it's going to be, to store all of that information. And then you've got to get contrast, labeling going on for you, and you need it in 3 dimensions. You got to be able to scan this thing in many different angles so you can get a 3-dimensional image of this in real time. They want 30 frames per second. So this is kind of, you know, the bar that we have to jump over, the super high jump that we have to do to really make important contributions in neuroscience and in fundamental bioscience. And it's possible. Here's an example of electron micrograph. I have been maliciously attacking that all along here, but showing a T cell coming into contact with another cell. This is the infected cell coming into contact with another cell. And what we need to be able to see, which the electron micrograph can see, but on a dead cell that has been sliced and diced and whatever, are these little buds here where the cells come into contact. And there's all kinds of proteins. Proteins are little, if you don't know about them already, they're little machines, molecular machines that can do things, very important things. In this case, they deform the membrane of a cell so that it can then grab a hold of proteins on the other, or in this case, cell that's going to get interacted with or infected, and open that cell. As soon as it's opened it, then it can inject proteins, viruses, whatever it wants to, from the disease cell into the healthy cell, and then spread the infection that way. And so the virologists have challenged us and said, I want to know how this happens. Because, of course, we virologists want to design technology that can stop it from happening, or pharmaceuticals that can stop it from happening. So we need to have resolution. Again, I told you the big challenge before, nanometer scale lengths, nanometer dimensions over millimeter scale lengths. In this case, could I just get 50 or so nanometer, maybe 10 nanometer resolution, and see these little buds and observe that in a living cell? So we've been trying to do that. This is how far we've gotten with what we call GFP, or it's a green fluorescent protein that is also embedded in HIV and can be spread from one cell to another. And I've showed you this image several different ways here. But this is a diseased cell, and there's a cell you can't see here. You can actually see it in this image. It's the same thing, only this one's colorized. So there's a diseased cell here in green. This one's not yet diseased, therefore it's not fluorescing. In this case, you can see a little bit better. And you see these proteins? They're being injected from one cell to another. And this little region here is called the synapse. And that synapse region is exactly this point. It's the point where those cells are touching. And it used to be thought that cells sort of exploded after a while, and then bacteria, or viruses in this case, would float around in intracellular fluid and then go infect another cell. Maybe that's not the case. What we're observing now is they have to come into contact with one another. That's the most efficient way for infection to spread. We have some very fancy microscopes that do this. It's called basically a confocal, spinning disk confocal microscope, which we'll show you more about when you do your visit of the center. We're also going for very high resolution. This instrument is something we built. I think I heard Marco mention it. This instrument can go to 5 times the diffraction limit of the light. So you can see, it used to be thought you could only see things that were roughly on the same dimension as the wavelength of the light that you were using. We can see 5 times smaller than that, which gives us the ability, using, for example, 500 nanometer light, green light, to see down to 50 nanometers or so in dimension. And that gives us incredible power to study living systems without having to use X-rays, which can ionize. And here's an example of this on some microspheres. The best microscope you could buy today, one that is now just been put on the market thanks to us and our colleagues at Applied Precision, and one that we're going to bring to market a little bit later, which has a resolution of less than 50 nanometers using 500 nanometer resolution. So, but to truly get down into the molecular scale of imaging, you're going to have to use X-rays, X-ray diffraction imaging. And this now is also brought to us, the possibility of this is now brought to us by having X-ray free electron lasers. There's a new one being built at Stanford. There's some that have been built already in Germany and so forth. And they're coming online as we speak. And we've been taking advantage of this, designing some experiments of this, because these things provide a tremendous number of photons per pulse, extremely short pulse duration, and very, very short wavelength, giving us the ability to take a single molecule, if you like, drop it in a beam of X-rays, and take a diffraction image of it, and then reconstruct that image into what the living, or in this case, the molecule would have looked like before we hit it with X-rays. Bear in mind, when you hit something with that many X-rays and that short a time duration, it blows it up. So you have to take this image very, very quickly. But that's how we're going to measure things on the, if you like, angstrom dimensions. We're also learning how to sort cells of different types, human embryonic stem cells, different stages of differentiation and so forth, looking at spectroscopy from them, light that scatters. Marco mentioned it earlier, talking about things, and one of you was clever enough to mention that at absolute zero, perhaps things don't vibrate too much, although there is some arguments about the vibrational levels of the universe as a whole, but we won't go into that. They're very slow. But the idea here is scattering off of vibrational bonds, molecules that vibrate under certain conditions. We can scatter light off of them and make them vibrate, and it tends to be able to identify specific proteins, if you like, the abundance of proteins and so forth in cells. And we've used spectra that we've done this way, where we've trapped a living cell inside a fluid, scattered light off of it, and identified different cell types. We can identify stem cells, which turned out to be very difficult in the past, to identify different types. Here's a movie of how this will work eventually with a flow cytometer. In this case, we have a channel, a fluid channel. You can see it here. I'll come back to the movie. But the bottom line is these cells are flowing down a channel, and there's fluid in here, so they're living cells. We shine light on them, we get a spectrum, and we say, aha, good cell, bad cell, the cell I'm looking for, the cell I'm not looking for. And then you can see here, I'm going to grab a blood cell. That's a big old blood cell. I pulled him over here. I grabbed him with a laser tweezer. I grabbed him only using light. And then I held on to him while I shifted this little microfluidic stage over and let that cell, the movie quit, let that cell, I won't start again, I'm sorry. We grab a cell, bring him over. See if I can see one more for you. They're big one. There's a big red blood cell. Grab him, pull him over, drop him. So that's the technique whereby you can sort cells. And you have to get up to where you can do this 1,000 times a second for it to be efficient. We can do it about once a minute, so we got some work to do. Okay, the last thing I want to talk to you about is future of biophotonics, and you can help here, I think. So technology for healthcare it definitely needs some revision. I love this cartoon from the past, especially when it's the complete physical one. You know, it's a little daunting to go from the simple blood pressure measurement to something that pokes holes in you and so forth. So the idea of non-invasive medicine, and I think light can get you there, is very important. So more seriously speaking here, the issue is the strategic application of medical technology can improve quality of life and cost effectiveness of healthcare. And what I like to show here is a quality of life curve. What we'd all like to do, and you're very young, so you're way back here, but we like to have it where we go along very high quality of life, and then one night we just don't wake up, or we get hit by a big truck or whatever. I mean, the best thing is that. The worst thing is going along and suffering, you know, some kind of disease or some kind of accident or whatever, which generally speaking, afterwards you have a lower quality of life. And then if you look at the cost of fixing these sorts of things, again, diagnostics aren't too expensive. Therapy gets very expensive and so forth, where it really have to throw the kitchen sink kitchen sink and things, which is where we tend to do medicine today. It gets extremely expensive, and certainly when you get very old, it gets the most expensive at all. So the idea here is accurate screening and early diagnostics can minimize healthcare. So I had to give this talk somewhere in Europe recently, so that's why I say a gram of prevention, but it's the old ounce of prevention is worth a pound of cure. So disease management through technology, and biophotonics is very key to this. I want to show you here. And one area that's the most exciting to me is prevention. So there's a gene chip system put out there made by Affymetrix. Do you guys still do a tour?
That's with the younger students.
Oh, okay. Yeah, there's some tour that Marco's done in the past with at Affymetrix plant here. But again, remember I showed you early on this whole idea of looking at a gene array? Well, this is how you can learn something about your own gene. And that gives you the ability to increase vigilance, do supplements, never smoke, exercise, and so forth, to improve your health so that your quality of life stays high until you hit that brick wall or don't wake up someday. So that technology and others has spurred a whole initiative now I'm involved in, we are involved in foods and lifestyle for health. The idea here is that your phenotype, who you are, is defined by your genotype. That's who your parents are and what they gave to you, your genes, and your environment. So here's mom and dad's chromosomes that combine to make you who you are. And then this is what you do to yourself. This little cartoon probably is not visible to you, but it says, I haven't got the gene, so it's fine for me to smoke. And this one says, I've got the gene, but I guess I'm doomed anyway. I'm doomed anyway. So whatever excuse you need, if you like to eat a lot, so forth, you can take whatever your fundamental good genes are, or bad genes, and usually make matters worse. And the key here is nature may have loaded the gun with your genotype, but you're the ones who pull the trigger with what you do with it. So from a technology standpoint, we want to give people the information they need. Now, there's a company in the Bay Area called 23andMe, and they have this work. They have stimulated these parties that people have now. You know, they used to have Tupperware parties or whatever. This one is spit parties. So this is where you spit in a cup, and then you send it off to have it genotyped. You have yourself genotyped. This is controversial. They look for something called SNPs on your genes. SNPs are sometimes well correlated with certain disease types. You know, are you going to get cancer when you're 55 or whatever? And there's a gene array. It's all based upon biophotonics, looking at these different colors and so forth here. There's another group called Genova Diagnostics, which is not so much focused on just getting your genotype, but also selling you, more importantly, all the drugs and supplements you might need to ward off the horrible diseases which are brought to your attention here. Some biophotonics equipment other than looking at arrays here, gene arrays, is shown here. This is actually something that's based on Raman scattering. This is what I was talking about earlier. I didn't mention that term, but when you're scattering light off of vibrational bonds and it changes the wavelength of that light, that's called Raman scattering, named after a famous physicist named Raman. This one is to look at antioxidant levels in your skin. So you put this little device on there, you measure antioxidant levels. If it doesn't look right, then Genova Diagnostics will be very happy to sell you some good supplements to take to make it all go away. This is a very serious one. This is a portable blood analyzer. So you can take a sample of blood, look at any kind of analytes in your blood that you're interested in. This is very important for an initiative we're pursuing now in rural medicine and in point-of-care testing. We just formed a new center inside of our center, rapid multi-pathogen detection for point-of-care testing and national disaster readiness. This is all based on pandemic medicine. This is, if you recall, Katrina, the big hurricane Katrina. What happened there that was a major catastrophe other than the flooding was that the flooding took out the entire infrastructure of the city, including all the hospitals. So what do you do in that sort of situation? Well, you need to build portable diagnostics that can be used to look for infection, all the other standard measurements or diagnostics you need for a hospital, but they need to be field portable. So we're developing, as part of the center, those technologies. And There's many of them I can show you here. Probably everything from sniffing the air to looking at DNA samples to even, this is a modified early pregnancy detector that is looking for whether or not you've been exposed to radiation, because a pandemic event could not only be a flood, it could be because somebody detonated a dirty bomb somewhere. And it turns out that there are biomarkers associated with how much exposure you've had to ionizing radiation. So in closing, just a few words about emerging healthcare technologies. Again, my favorite cartoons here. We hope to get away from something that looks like an electric chair to try to determine your overall healthcare. And one of the things I'm most interested in is how much can we use a cell phone as a telediagnostic? And already there are people working on this from UC Davis and UC Berkeley, where they take pictures with a modified lens on your cell phone, turn it into a microscope, so that if you're in the Sahara Desert, sorry, more likely in sub-Saharan Africa, in the jungle somewhere, and you got bitten by a mosquito and you think maybe you've got malaria, they take a sample of your blood and they take an image of that blood sample, send it somewhere else for analysis. That's called telepathology, and that's what's going to happen in the future. Now we want to expand it. We want to measure blood glucose level, and so forth. As a matter of fact, this is an example of it called a glucophone, where they take a standard glucometer, take a sample of your blood, and then that information is analyzed and then sent somewhere to your primary care physician, and so forth. Cell phone is a telemicroscope I just mentioned. We're putting together right now, I'm going to talk to somebody, matter of fact, in an hour about this Institute for Rural Medicine and Innovation. How can we implement medical care for people who don't live in a big city, so that everyone gets the same technology, everyone gets the same quality of care, no matter where they live. So, with that, I'll shut down my lecture. This is how you get humiliated when you get to be 60 years old. And these are many, many people, students who were at our last retreat in Lake Tahoe. So thank you for your attention. If you have any time left.
Thank you guys for staying a few extra minutes, and we'll see you again on Wednesday.